also be looked into so that we can be able to know based on the priority crops that we have. Thank you.
Yeah, I'm Patrick from Narok. What was interesting for me is that I realized from the presentation that we have crops which you can grow, the trapping crops, to discourage multiplication of nematodes. So in case of management, other than Velum, as we said, that we provide solutions, so we can be able to advise farmers on different ways of managing nematodes. And also realize that there are so many nematodes. We have been very skeptical that we don't have so much nematodes in some of our areas, but we see we are very careful and we do more research. We will find that we have a lot of nematodes to show the farmers what to do on those nematodes. Thank you.
Thank you, colleagues. Quite interesting comments, and yeah, looking forward to the practical session. So I want to hand over to our researchers to guide us in the next session. Miriam Kungu, she's going to guide us into the practical session, possibly guide us on how we are going to group in around different tables for the next, I'm sure, 1 hour or so. Thank you.
We've come to a time where you're going to see physically what our professors have been discussing this morning. It was a very interesting session, and we always have, even ourselves, after being there, we still learn a lot from them. And we are happy that this has generated quite a good discussion. So from these people you see here, they are not medical doctors. If you're afraid of needles, they're just wearing white coats. They're actually plant doctors, and they are going to take you through the bridge. They're the bridge between the field where you are based and the laboratory and the research section. Basically, they're the technical team composed of students, PhD students, masters, and technical team who are experienced in handling the nematodes from the farm, bringing them to the laboratory, processing them, and finally identifying so that we can provide the management practices that are required to increase the yield in the fields. So we have 4 tables. It's not easy to do. It wasn't easy for us to be able to do all this, the field, the laboratory, and management in 4 tables. But what we're going to do, we have the far end there, which shows the farm. It's like our farm, because we have the posters which are going to show us the symptoms, and some plants material there that are going to show you the symptoms that we are talking about. As we are told by Prof, nematodes are in all crops, but we've selected just a few that are maybe of more interest in this country concerning the type of nematodes you are told, the banana nematodes, the root knot, and the potato nematodes. So you're going to see that from that corner. There are the posters. We have the plant material. Then, do you remember after the symptoms and the problem nematodes cause, there was a discussion about sampling? Because you need to bring this this soil. We are actually mostly there concentrating on the cell nematode. Maybe we'll be told much more about sampling of the leafy nematodes above ground. So you're going to bring that material from the soil to the laboratory. So you'll be shown in the next slide how to sample. The next table. You proceed, and then there'll be a table where you'll be, the second table. Once you bring this plant soil material to the laboratory, after you've been shown how you sample, what do you do with it? You have to process. So there's a table on extraction, how you extract your nematodes from the soil. You'll be shown how to do that, depending on the type of nematode that you've come with. In one soil sample, there could be different nematodes, as you've been told. Some may not even be parasitic, but they're all going to be processed in the next table. Then you'll be shown how you concentrate that to make sure that you have enough nematodes that you can bring to the table and identify. As you do this, you're going to have started your first step towards learning nematodes, like taking baby steps. It's a big thing. We keep learning every day, but this will really introduce you to the whole process, Maybe a farmer will tell you to sample their farm. So what do? You can process a bit before maybe you bring to the laboratory, or at least you can know that the researcher will do that. Here you'll be shown how the nematodes look like on this third table from that side. On the screen, somebody will be sitting there to show the different types of nematodes. After you do that, after you have the nematodes identified, of course, you move to the next step, post-identification, where you do management. We didn't bring all of them here, but some nematodes might need to be multiplied, cultured, like the plant parasitic nematodes. So there'll be a table here showing you how we do laboratory culturing. You may not do this in the field, but you might find yourself doing it at one point or another. Then there'll be some discussion on how we have been doing screening the different nematodes for the potatoes, for different nematodes for resistance. There are other procedures we didn't show on management, but this is just basically what we do. One of the things that can be done once you identify the nematode that you have, you can culture them and continue testing maybe on your Velum to know whether which dosage are working or something like that, and the screening. So we start from there. We may not be able to divide you completely, but what we need, we are still thinking whether we get into 2 groups, but the best thing is that you start from there. Don't jump here because you may miss something. Make sure you understand the symptoms for that table. Make sure you understand how to sample, how to extract, how to identify, and then finally you come to this table. Prof, is that all right? Okay. So we could start from the one sitting here. Yeah.
How many tables do we have?
We have 4 tables.
4 tables?
Yeah.
Which is the first step?
The first step is the symptoms there.
Is the symptoms.
Yeah.
And how many are we? Can we say 5? Group of 5, 5? 6? 6, eh?
Some sit down, I think.
Yeah.
5 do.
Yeah.
As they proceed, the other group joins in.
Yeah.
As you proceed, once we all have seen, then you can settle where you're more interested.
So we can have these first 2 tables, start from the end, and then when they move to the next table, we are the next, right? Okay. And Dr. Solvay wants to say something because I think you're leaving, right? Yeah. But
Hello, everybody. I just want to say, unfortunately, I've got to leave you, but in the good hands of people here. But it was great to meet you, and we definitely will meet again, I think. And feel free to contact us. Diana has our contacts, and sorry, I've got to leave right now. But thank you very much. Enjoy the rest of the day. I'm sure you will learn even more, I promise you. Okay, thank you.
thank you so much.
We are going to showcase you nematode symptoms on 3 crops. On potato, that's the PCN, and then spinach here will be representing the vegetables, and then we'll have banana. Although there are other crops, but we chose out these 3 just for your demonstration. So usually when you go to the field, you would be seeing this very nice looking potato crop. Most often you may not be able to tell whether it has nematodes on the roots until when you pull it out, and then you will be able to see actually the cysts on the plant itself. Before I show you the cysts on this crop, if you look at this image here, these white balls or dots you see here are actually the PCN, the protocysts on the potato crop. Now this plant was sampled by one of my colleagues 2 weeks ago from a potato farm. But if you look critically, you see these brown dots here? They're very brown. These ones here. They're here. Okay, these are potato cysts. So this is just going out from the field, and then immediately he could see the cysts on there. They're this brown, you see? So these are the potato cysts, yes. Don't take that one to you. Yeah, red, yeah, yes, they're here, yes. Usually when they are still young, they're white. Then when they grow and then when they're almost hatching out, then the cysts turn from white to cream, then to brown. So these are already mature cysts. Now in there are so many eggs of potato nematodes, which will eventually hatch and then infest more new and new crops. So these are like the examples of how they would look like. See, it's there. That's a system. That's a, yes. Actually, these are attached onto the plant. Yes.
You may find that in different colors.
Yes, you could find like the white ones, the cream ones, the brown ones, because they have different developmental stages. So some might still be white, some have turned into cream, while some have turned into brown, like what you've seen here, because most of these are golden brown actually. Now these are the mature ones. But even the cream and the white ones, if you pull them out, they already have eggs that can hatch and be able to infect the next stage of the crop. Actually, these cysts are now like the hibernating stage of the plant. These can stay in the soil for years and years. Even when you don't plant potato for the next year, 2, 3, 4 years later, if you plant, they will still be there waiting for the host, because they only hatch when the host plant is there. If there's no host plant, they will remain dormant and docile. When you put your potato there, then they will hatch and then infect that potato.
So how did you suspect all right. How did you suspect that you could be having nematodes infestation, considering the foliage?
Looking on the leaves, you may actually not be sure if it has nematodes or not. Best is actually on the leaves, it might look very green and looks healthy and all that, but it's only until when you pull it out, then you realize you have cysts on there. Yeah.
Maybe a question. What was the stage of this crop when you were taking the sample? Was it the same size with this one, or it was
the stage of the crop?
the stage, yeah.
Usually,
This specific one.
yeah,
I'm
usually,
asking this specific
yeah.
one.
I'm not the one who really sampled this. It's a colleague of mine who sampled, but usually at the beginning from the flowering stage, you can be able to see, yeah. From flowering until harvest, you can be able to see.
Now, from your view, in this plant, do we have foliar nematodes, or how do we identify them?
No, this specific one, they are not foliar nematodes.
How can we tell? How can we tell?
How can you tell? Usually for the potato plant and foliar nematodes, maybe Dan can help out on this, but there is not so much foliar nematodes that attack potato crops.
There's a huge amount of interest in foliar nematodes. I think the thing to think of is, by and large, most nematodes are root parasitic, basically. And then there's a few... Sorry. Oh, face the camera, sorry. And then... Oh, he asked a question. And there's... mainly the root parasitic nematodes and in the soil, but there are a few that will cause damage to foliar leaves and stems, basically. They are a minority in comparison to the root and soil nematodes. And the way to find them on the leaves is that mostly they cause distortions, but also they cause discoloration and chlorosis and chlorotic patterns. Whereas a disease, you'll see that it's disease and it's necrotic. Whereas nematodes tend to cause, it tends to be more straightforward, basically. Yeah, it's not diseased, it's not powdery, it's not smudgy, it's different colour. Yeah, okay. But interesting if you find them, because we don't see very many. But we did have a report of some in the chrysanthemum farm, but we never got samples to confirm.
Unless if there are any more questions about PCN, we can move to the next nematode, which is what Danny focused on mostly. That's the RKN, or the root knot nematode. This nematode attacks almost every crop. right from those with the soft roots, the vegetables, to the grasses, even the trees. Can one of you tell me what this crop is? Spinach. So if you look at, this is how a clean spinach root system would look like. I mean, no galls, no anything, very well grown, main roots and feeder roots. And then, if you look at this, although the roots actually look big, bigger than the other one, but what you're seeing here are the galls being caused by the root knot nematodes. So this is like a sign. Whenever you go out in the field and then pull out that plant and you're seeing these galled roots, then you know such a crop is infested by potato cyst. Not nematodes, root knot nematodes. Yes, because some other nematodes don't cause the galling, but the galls are due to the root knot nematodes specifically. Yeah.
What happens to the galls? What happens to those galls? Do they erupt afterwards? And what happens to the nematodes that are inside?
Okay. Now, in each of these galls is the adult female. So it lays eggs. You may not really be able to, I think you can. If you see here critically, now that is an egg mass containing the eggs laid by the female in this gall. Actually, these galls are like cells onto which the adult female is feeding. So as they expand, the roots also expands along.
You can see that under the microscope as well, James.
Ah, yeah. So in these egg masses, there are the eggs of the root knot nematode, which will hatch, and then now move onto a new crop. Yeah.
Or a new root.
Yeah, one new root, yes. Any more questions? Okay, then we move to the other nematode, the banana. Who of you guys comes from a banana growing farm? Okay, before I take you to this, just look at these pictures here. You have this picture and you have this picture. Oh. Which of these is good? Why do you think this is the best?
Greener, no dead leaves.
Okay, greener, no dead leaves, yes. But then if you look here, you're seeing many of these plants, they have fallen. And this is because the root system of these plants was infested by the nematodes, so they ate away all the roots, so the plant cannot anchor itself into the soil. So the plant toppled, and as a result, your field looks like this. Usually you go to the field and then you find some plants when the comes remains in the soil and then it breaks at the soil level. That is a different pest. For nematodes, the whole plant uproots, just like you see here. But in the case, like the plant just breaks at the soil surface level, then that's due to banana weevils. But for the nematodes, the whole plant uproots because the plant no longer has enough roots to anchor it into the soil because the nematodes have eaten away the whole roots. So it falls over. And actually, some of what is here, you can still see them here. If we look at this first plate here, on the surface of the banana root, this is what you should see, very clean and no damage and all that. Even when you slice the root into two, you see at least the sliced part of the root is clean and healthy and all that. So this is how a healthy root, banana root, should look like. But then the infested one, even on the surface, you already see these dark and brown patches on the root. So when you slice it into two, you see this necrosis on the sides. Now these are portions which have been by the nematodes, yes. So this results into toppling over the banana plants, and before you know, your field is all destroyed. Now farmers, what's the usual or most common way of planting bananas? Those who come from banana growing, how do you establish your new suckers? A sucker like this one, which obviously you see, one, it has no roots because they've been eaten away by the nematodes. And then even when you, although, okay, don't focus on the tunnels, these are the weevils. But still, when you look at, you see these brown and dark patches, those are already nematode yes, areas. Although you can try clean this by pairing it, peeling it off the outer part and then dip this into boiling water. It might help and clean the sucker free of the nematodes, but still, when you plant this into an infested field, it will still come back. It will still be infested by the nematodes. Same applies to tissue culture plants. This looks small, but actually this is clean and free of any nematodes. So planting this in a field, you'll at least have your field free of nematodes for the first cycle, second cycle, and if there are no nematodes, then the field can last for some time. However, challenge with this too is in the case that field is infested with nematodes, still you will have them also being attacked even when you begin with the clean planting materials. So that's when the other management options come in, like the endophytes that you can use, like the nematicides, although nematicides now have their also downside of being harmful to you the user, and again also to the environment. So looking at the environment friendly alternatives like the endophytes, which is what I'm basically working on, is a promising way of how to manage these nematodes in not only bananas but even other crops. Although my question to you, I see today you're promoting the use of Velum in potato. And maybe, I don't know if the same can be tried out and tested on even other crops, and if it can work. That could be a research area for you guys and maybe us as well. Yeah. Okay.
A question,
The
hot water treatment.
Yes.
I don't know how the process goes by, because maybe I can subject the same nematodes on very hot water, yet I'll be killing the cells in the same plant. I don't know whether you have a specific degrees on the water treatment process, or else it's just putting them in hot water and then does the process take, maybe you can take us through the process. That's the question.
You're very right. If you expose this plant long to the hot water, you end up boiling it, so killing it. So the best way, especially for the farmers who don't have the equipment measuring temperature and all that, the best way to implement this is have the water boiling and then dip your paired sucker in the boiling water for 20 seconds, not more, not less. If you put in for less, it will not kill all the nematodes. If you put in for more, you will cook the plant and it will die. Yeah. So 10 seconds boiling, yes, it's the best.
The other question is, the hot water treatment, does it has a capacity even to kill the eggs on the same galls, or maybe you can elaborate that?
Usually these nematodes, when they lay their eggs, it's on the surface of the comb. So by you pairing, you're getting rid of some of the eggs and the juveniles, but whatever remains will still be on the surface. So by you dipping the sucker into the hot water, at least it will be able to penetrate a portion of the comb that contains the eggs. So yes, it will kill them. Yeah, because the nematodes don't go deep into the comb of the plant. They deposit their eggs and the juvenile hatch on the surface of the comb. Yeah, actually most of them lay their eggs and they hatch into the roots, which you already
On the surface of the root or inside the root?
Usually it's the surface, but the roots are like soft compared to the comb. Yeah. So as they hatch and then they begin eating, they tend to go into the root. But still, if you realize here, the inner part of the root is clean. No, not this. The inner part of the root remains almost clean all through, but then the outer surface, the cortex, is the one that they have fed on. So they feed onto the outer surface, and then the inner part is left intact.
The
And
hot water treatment will be able to reach to this point.
yes, it will. Yeah.
Thank you.
Okay, then I can hand over to my colleague.
Okay, now we are at the sampling stage. From what James has discussed concerning symptoms, for you to sample, there must be a reason why you want to sample a certain field. It could be maybe a farmer has complained of a low yield, or you could have noted that there are low yield, there's low yield in the farm, or you could have noted some patches in the field, or you could have someone or a farmer might be wanting to plant a new season and might want to know the level of nematodes. So before you go for sampling, you need to plan for it. The time for sampling, of course, is dependent on many factors. It's not good to sample when the soil is not too wet, neither too dry. It should be. And then when you're going to the field, there are tools you're going to use. You need a cooler box, not necessarily this size. It depends on the number of samples you want to bring. And that also might depend on the reason you're sampling and the size of the field. That will determine the number of samples you might want to carry. So you carry a cooler box that is, because as you were told in the morning, we do not want to expose our nematodes to direct sunlight and high extreme temperatures because they are living organisms. So in the cooler box, you're going to have cold packs, the ones that we throw into the cooler box to make sure it is cold. I don't think I have one here, but you know. So then once you have that, you're going to have your shovels. We need shovels. And actually, I'm discussing basically all these types of nematode sampling, and if there's something to add, we're going to add. The method is basically the same. So you need shovels. You might need an auger. This is the auger. You might need a knife because when you are sampling, you need to carry soil material. Soil and also roots. Some roots can be thrown into the soil so that because some nematodes are in the soil. You're going to have a packaging material. You could use a khaki bag or even a container, a plastic container. You're going to have your labeling tools, like you're going to have a permanent marker, a pencil also. If you're using a label like this one, you're going to write using a pencil because it doesn't come out easily. A permanent marker, possibly for this paper or the box. Then you find yourself in the field. What do you do? So you look at the size of the farm. If it's a very big farm, possibly you could divide it maybe to hectares if it's a very big farm. But the sizes we have in this country, they are smallholder farms. You physically divide the farm in, not that you take tapes, you're going to visualize your farm and look at that farm. Do you have visible symptoms or is just a crop that looks even healthy? So then you choose the pattern you're going to use to sample because we have different patterns. If the farm has patches, you could use a sampling. We have basically two sampling patterns. We could have random or systematic. You basically use systematic because it's more focused. You can use it even when you're not sure whether you have nematodes or not. But sometimes you might just want to sample a patch area. You might use a circular method. A circular method is that you might have a visible mental circle, then you're in the middle, and then you move towards the edges, like you're making a star. And then what you do when you're sampling, what we use mostly is the zigzag. We also have a zigzag method, which we use mostly for any crop or even a farrow farm. Maybe there's no crop growing. So you visualize your farm and you move in zigzag manner. As you take scoops of soil at the depth of 10 to 15 centimeters. But first of all, you have to scrub off the surface soil because it could be exposed to a lot of high temperature. You scrub a little bit of the topsoil, about 5 centimeters deep. Then you scrub. Then you go 10 to 15 centimeters deep near the roots of the crop, and even in between the rows. You move in a zigzag manner, depending on the size of the crop, of the farm. You can take 20 samples, and then you combine them in this bag or the bag you're using. So those samples will make one composite sample, which normally we take 1 kg to the laboratory of the soil. But you don't pick in one area. You move in a zigzag manner, picks 20 scoops, which are sub-samples, and then make one composite sample. So the size of the farm will determine how many samples you need, and possibly the reason why you're also sampling. So as I said, you're going to use this shovel. You might also use an auger. Maybe you want to pick samples from a place that is a little bit deep. You're going to move to push it down, and then you to a level that you can be able to pick the soil, and then you're going to push the soil out, put it maybe in a container, and then you throw it in a bag. So then you're going to put your soil in the cooler box, cool it with a cooler pack. And before you put, don't forget to label, very important, because the researcher in the lab there, that researcher there, who is working on identification, will want to know what crop it was you collected from, what region it was you went. So you need GPS, you need the crop. You might also want to know the stage of growth of that crop. You might also want to indicate the farmer if necessary. And maybe you give it a code. GPS, did I say that? And the date also, don't forget the date, very important. I think, yeah, those are the materials you need. That's how you label. You might also, as you move from one field to the other, especially when we are sampling for PCN, it's a quarantine pest, you might need to clean your shovels. You clean them so that you don't move pests from one farm to the other. And then, you maintain that quarantine status of the pest. Any question?
Why do you put it in a cool box?
Okay, you put in a cooler box to make sure that the sample doesn't dry up completely and the nematodes do not die. You don't expose them to too much high temperature. And actually, when you bring them to the laboratory, if you're not processing them immediately, you can keep in fridge, maybe 10 degrees centigrade for even 2 weeks or more. 2 weeks. If you keep for long, they might deteriorate. So it's to make sure that you bring the sample in proper status, that the nematodes are still alive. And especially depending on what type of nematodes, when you move to extraction, they'll tell you some you might need it most, the soil, some you might need it dry, depending whether it's PCN. Okay, maybe Prof can add something on that.
Yeah, I was going to say there's two of the simplest things are the things that cause the biggest problems. One is labeling, okay? It's so easy to label, but it's also easy to do it badly or not at all. And it's so important to establish which sample is from where and for which reason. And the other one is storage. Don't leave it in a boiling hot car in the sun while you go and have lunch. Otherwise, everything's dead, okay? So there's so much effort and time spent sampling, and they are destroyed in so little time. And we did a study with a particular company recently, and we discovered that 90% of the samples that they were getting, they were spending money and charging the client for identification of nematodes, and they were saying, you've got no nematodes, no nematodes, no nematodes. Okay? Not only is that a waste of money, but it's completely inaccurate because the sample they were all dead because they've been left in the sun, they've been left too long, they were not labeled correctly. So the two simplest things are often what are the biggest problems. So storage, take good care of your samples, and label them properly. Okay?
So does it mean from when I take the samples to when I send them to the analysis, we are supposed to use the cooler boxes? Because normally we have used these bags, then we send them via Fargo to very far distances. It means we didn't get the actual results.
Yes, ideally you need the cooler box to avoid deteriorating the sample and the nematodes dying. You need to keep them cool, and that's as Prof has said, don't even leaving them long. But thank you.
They don't have to be in a cool box, okay? If you're up in Kinangop, and the sun's not shining, they're okay in the car. And if you're out in the field for 3 or 4 days, if they're in your hotel room or you have an office, on the floor, in a nice coolish conditions, that's fine. But if for example, you're out sampling and you go for lunch and I'll pick them up later when I come back, okay? They're not going to be very good. Your nematodes are going to be really, really unhappy, okay? And dead, okay? And a dead nematode is an unhappy nematode.
Yeah, that's well answered.
Cool box are useful, but common sense, just keep them in a reasonable temperature and condition.
Yeah, if you don't have a cooler box, it's an option. Don't carry them in the car. Leave them in your room, possibly, or somewhere cool. Any other question? Then we can move to the, after you've taken the samples, you go to extract. My colleague, Onesimus, will continue.
So welcome. After you've gotten the sample, now you proceed to another stage where you extract the nematodes from the soil. And what I will take you through is the extraction of the potato cyst nematode. So for those of you who are working with the potato value chain, this is very important for you. So now I will illustrate two methods. Method one, the Fenwick can, and the bucket method. The bucket method is mostly applicable in the village because they are available, because Fenwick can would not be available in the village. Now, once you have the sample, Miriam said that some samples you need them wet and other samples you need them dry. Potato cyst nematode is one of those nematodes that you need the sample, you need to eat dry before you start extracting. Because once it's dry, the soil will disintegrate and the cyst will be released from the soil structure. So therefore, you work with 200 mils or 100 mils of soil, depending on the protocol that you have, and after you've received it, and you dry it away from the sun, so that the nematodes will not denature. So therefore, you scoop. If you're working with 200, you do 2 scoops of this. If you're working with 200, you do one of these. And then you're going to put it, you need 3 sieves. 1 millimeter sieve, 850 micrometer, and 250 micrometer. And they are arranged from the biggest to the smallest, so that you'll eventually have the cyst trapped at the bottom sieve. So what you do, you put the soil on the sieve up here, and then you need running water. So as you run the water here, it will trap all the big particles that are bigger than 1 millimeter, the rocks, the big debris, the plant organs, and the roots will be left here. And then after that, all that is filtered, the filtrate will enter into this barrel. And in this barrel, the soil settles at the bottom, and the cyst and the debris floats. So as you add the water, you'll eventually find out that you have something like this, where you have got the soil at the bottom, and up here you've got the cyst and the debris. And as you continue to add the water, it will flow out. And what flows out is what is floating. So eventually you have the debris and the cyst moving out of the barrel and the soil at the bottom. And then as you continue to allow water to come in, all the debris will come on this sieve. And then you have flowing water, and allow all the smaller, all the debris to remain at the top and to the top sieve, and then the smaller cyst will go into the lower sieve. So eventually you'll have the cyst and the relatively smaller debris on this sieve. Then now what do you do when you have it here? You will have to backwash it. If it is contained in here, you have to draw it to one of the corners and then backwash it. And then leave it for 5 minutes to settle. And then after the settling, there will be debris that will go at the bottom. And the cyst will be left floating at the top of the water that you have in inside the container. After that, you have your milk filter paper. You fold it into 4 quadrants like that. So by that you are creating trapping, and then you use this funnel. See this? And then assuming that the cysts are like this, you will now dispense them like that. So what you're left with here is the debris end and the cyst. And then we can take this and place it on a paper towel for it to dry overnight. Yeah. So if you look at this, there is a lot of mixture of debris and cyst. So there are some other advanced steps that you do to make, especially when you're working with many samples, to reduce the amount of debris. So after drying, you can put this in organic solvents, like maybe ethanol or acetone, with a lower density than 1.0, probably 0.9 or 0.8. By that, all the cysts will come at the top and all the debris will go at the bottom. And therefore it will be easy for you to extract it. And the bucket method is just the same. Just take the soil sample, put it inside the bucket, and then add water, and then it will be floating at the top of the bucket, like I did show in the previous extraction with the fenwick. Yeah. So with that, probably I would handle a few questions if there are any.
The extraction in the bucket.
Yes.
Yes. How do you go about it?
So when I did say that if you are using the bucket to extract, you just take your sample in the farm, dry it, and then once it's dry, take some 200 or 100 cc of the soil, put in the bucket, add the water, and then all the debris in the cyst will float at the top. It will not be possible for you to have the sieve in the village. So maybe probably use something like a muslin bag, you know, the inner lining of clothes. That would help you maybe to filter out. It would simulate the milk filter paper in the farm setup. Yeah. Thank you.
Okay, I'm Celestine. So as you have seen from sampling, you come to extraction. So we have different types of extraction methods, and one you've seen, there is fenwick can, and that one will only, it is used to extract cyst nematodes. So we also have other methods, like centrifugal flotation method. We have funnel method, and then we have modified Bayerman technique method. So why do we go for this? First of all, it is cheap and it's affordable and very easy. Like anyone can afford to do this. You only have this. This is around 10 shillings. You use this one. Then you have the serviettes, or you can use Kleenex. So when you come here, we have 2 types, like we want to extract from 2 different media. We have soil and we have roots. So I'll start with root. So the first thing you do, you have your roots, and you are told like labeling is very, very important, because like, for example, I come with this and I didn't label it. You can go pick another sample, you just find like you've mixed everything. So it's very, very important to label, and it's good to use pencil, because it won't rub, like maybe you are in the field, there is rain, you use ethanol. If you use pencil, it's the best. And it's good to label the date and the collector. So after that, you come with your sample in the lab or extraction room. So we have our roots. So the first thing, you wash it in a running tap water. So after washing, you come and cut it. We just use these simple things. So you cut into pieces, into smaller pieces. Then you pick your plate. So after picking your plate, we have this sieve. Then you just place that here. Or you can come place okay, sorry, it's roots. So after getting this one, you just put it there, then you tear, because it has some weight. Then you come and place this 5 grams. Yeah, because you want to quantify at the end of it. You can take it back to how much roots you had. So you just weigh 5 grams. After getting 5 grams, you put water gently. Okay, you should not put a lot of water, because nematodes are water loving, they're aquatic in nature. So if you put a lot of water, they will all float up. So you should put at least around 200 ml of water. So this one will allow the nematodes to move from the roots into the water. So it will take, you can leave it for maximum collection, you leave it for 72 hours, 48 to 72 hours. Then after this, after 70, okay. After 72 hours, sorry, you just come with your sieve, and the sieve we are using here, we saw that one we are using 1 micrometer to 250, but here we are using 25 micrometer sieve. So 25 will help to trap all the nematodes that are in here. So after your 72 hours, you just come and remove this gently until the water is like there are no more drops of water. Then you can discard this and make sure, remember there are nematodes here, so you discard it in a very nice place where you will not interfere with, you will not spread the nematodes. So after getting this, okay, remember also there's labeling. You have to label all your samples, because you may confuse them or you may lose them. So after this, you just come and concentrate it here. Then you wash the plate, because there are some nematodes, maybe they are there. So you just wash the plate, then you concentrate it here. After concentrating, you come with your Falcon tube, and then you just pour it inside here. And you can, again, rinse 3 times, 2 to 3 times, to make sure that you have all the nematodes. So after this, you leave it to settle overnight. Then you can come and reduce your water. Then you go with it in the microscope. So the same with the soil. When we come to soil extraction, you have the same setup. Then you take 100 cc of soil. This thing is usually labeled. At times we make mistake. We put it up to but it's labeled. It's 100 here. It should be somewhere here, not at the top. So after this, so for soil, it's a bit tricky, because if you pour water before putting the soil, you find that if you put soil before pouring the water, you find that it becomes very, very dirty. So the best thing, you put this first, then you put water up to around somewhere here. Then you come with your tissue, you place it there. Then you just, this is 100 cc, you put it there. So if you can, okay, it's approximately 200 ml of water will be enough. So you leave it for 72 hours, 48 to 72 hours, then you come, you do the same procedure. Yeah, so it's good you leave it for around 72 hours, because some nematodes, like larger nematodes and larger nematodes and slow-moving nematodes, they take some time before they move from the soil to water. So it's important to leave them for that for 72 hours for maximum collection. Yes, maybe if you have any question.
What if it's hot and dry?
If the environment, yeah, you can cover them with a cling film. It will help to reduce the transpiration rate or the evaporation rate. Yes, so the soil will remain dry, and the nematodes will be very, very happy because there is no distraction above, so they will just move to the water. No, if at all, let's say, okay, it's a nice question. Let's say that you left your sample and you find it's dried. It dried up, so you can just come. Make sure you don't interfere with this, because if you, like, what have I done? The soil, like, the sample is now dirty. So if you want, you don't interfere with this. Just leave it the way it is. Then come with your water. Then start adding from this side. Yes. Mm-hmm. Okay, for this method, you will just have to wait. But let's say, for example, you are using centrifugal flotation method, you'll have to add some sucrose, yes, and to make them float on the water. But this is very effective because anyone else can use it. For centrifugal method, you'll have to have a skilled labor. You have to have the centrifuge, and you know how much it costs. So, and this one, when you're having a lot of samples, like for our case in a research institution, you can just place it there, then you go and do your other work. Then you will come later, then you collect your nematodes. Yeah.
Why are you putting water from the bottom instead of the top?
Because if you put from the top, first of all, our main aim is to make the nematodes move from soil to here. So if the first thing, if you put from the top, all your sample will be dirty. Again, if you put from the top, most nematodes will come on the top. So when you will be removing this, you will be throwing all the nematodes that are trapped here. Yes. Okay, thank you.
It's simple and basic, but it works. It's not 100% effective, but it's good and it works. Give or take, yeah. We're trying to improve it. But it's consistent across places. Especially if you're on safari and you're sampling and effective, you can do it as you move along.
Good. So I just repeat the question.
Yeah, if like he asks why you put in from the top, you say we're putting from the top because.
Yeah. I don't know where to stand. Let me stand here. Can I stand here? Okay, so in this table we are going to show you the morphological identification of nematodes, and in this case we are going to look at the potato cyst nematode as well as other types of nematodes. Okay. So before we do that, I'm sure you were shown how galls look like, and you were told that inside the galls we have the females. So we have prepared a slide here, and you can see the female. Yeah, go ahead. I don't know if you know how to use it. Yeah, so that I don't know if you can see. You can adjust the focus here. Is it visible? You can see something like this. Yeah, so that is a female, and so inside the galls you find those kind of organisms. That's a female of a root knot nematode. Yeah, so you can go ahead and. Yeah. Yeah. So it has sort of like a neck, something like this. Yeah. And it's white in color. can see, yeah. Yeah. You've seen it, right? Okay. Oh, yeah, go ahead. You can use this to focus. The other one. Is it okay? Okay. Okay, great. So that's what a female of a root knot nematode looks like. Okay. So now, after extraction, I'm going to take you through the PCN. So after extraction, the first thing we do in the lab is quantification. This means counting your nematodes, knowing how many nematodes do you have in a specific quantity of soil. Okay. So now, once you quantify, now you know whether your farm has passed the threshold or is below or is within the threshold of high cyst contamination. So now, for counting, we use a device called a tally counter. I don't know if you have come across that. It's on page 62. You can see there are different types of tally counters. So there is one that counts has different counts different nematodes at the same time, while others you just count one nematode at a time. Okay. Now, once you count, you now sometimes in the soil debris it's hard to differentiate between the spores and the cyst. Sometimes you might confuse. So now the feature that differentiates them is that the cyst has a specific neck, sort of something like this, but I'll show you under the microscope. But it has a sort of like a neck, and it is usually golden brown in color. So let me show you how the cyst looks like. So here I've prepared a number of cysts, and you can go ahead and you can see those features. Like you will see a neck, and the color is usually golden brown. You can use this to focus. Is it visible? Yeah. So can you be able to see the neck? Okay. And the golden. Yeah, it's small. This is adjustable. Yeah. We also have tubes full of cysts, so you can also see them with your naked eye, but it might be impossible to see the neck, but you can see them. You can just pass it around. You can notice the golden brown round things. Yeah. Okay, so inside the cyst we have eggs and juveniles, and that's when you plant your potato. The potato usually releases exudate that stimulate the hatching of the eggs and juveniles, of the cyst to release the eggs and juveniles. And then now
How many eggs per cyst?
Sorry?
How many eggs in one cyst?
Okay, in one cyst we might have up to 2,000 eggs and juveniles. So you can imagine, like, when one cyst bursts, how many plants can be infected within a field.
So in each cyst, there's about on average about 400 or 500 eggs, and the same for the root knot nematode, about 400 eggs. Female, so one nematode gives 400, and in that tube of there's cysts, how many cysts do you reckon? How many? I want you all to guess.
1 million.
Not quite.
Or 200?
No, not
200?
really.
200 cysts. How many do you think? How many cysts in that tube?
Over
300.
Over.
Over 300. Chef? How many? 2,000? 5,000. 1,000. In that tube, don't know, but probably 6,000. And each one has got 500 eggs.
Now, as we continue, I have a question.
So yeah,
Yes.
so 3 million eggs.
Yeah.
When
Okay.
you receive a sample, how do you know which nematode will you analyze for? Like now, there is a root knot, there is PCN.
Okay.
When I submit a soil for sample, for nematode analysis,
Yeah.
which one do you analyze for?
Okay, first of all, we are guided by the crop you are planting. So, for example, let's say you had potatoes in your farm. The first thing we will look at is PCN, definitely. But now let's say you have tomatoes in your farm. We're going to look at root knot, nematode. So the crop is what guides us. So it guides us to know which nematode to look for. In other cases, in most cases, apart from PCN, we usually look at all plant parasitic nematodes, because you find that one crop might be a host to different types of nematodes. So sometimes we look at different nematodes. Yeah. Any other question? Okay. Ngoja maliza. You can use this. You can also zoom in. Unaweza zoom in. So focus and zooming in. Okay, if there's no more questions here, you can proceed to the other microscope.
Okay, yeah. This is Calvin's. I would like you to now see the real nematodes physically when they're moving so from extraction, where Celestine has extracted the nematodes, they now come to the lab. Let me say they're well labeled from that side. So I have a sample, but the first thing I do when I come from the extraction room from that side, I decide which is maybe the quantity which I should use for quantification. So I can decide that I'm going to use maybe 10 ml. Yeah. So I allow my sample to settle for maybe 30 minutes. After 30 minutes, the sample has settled. The next thing I do is just to reduce it to the quantity, the rate I'm going to use. So what I do, okay. What I reduce the water to the rate, the standard rate which is maybe being used. Yeah. So I have my sample here. When I'm reducing, I just put my pipette towards the touching the wall, almost the wall, so that I ensure that I don't suck my nematodes. I don't lose them, because I'm going to discard this. So while reducing, okay, the rest I discard to the quantity I'm going to work, with. So that the quantity I'm going to decide to work with is very important, because I'll be able to, it will be able to tell me what is the approximate number of nematodes, maybe per certain quantity which was extracted there. Okay, so I've already reduced my sample to 10 ml. Then the next thing is that I come under microscope, this one, and quantify. Just pipette, let me say I use a standard rate, 2 ml. I just standardize my pipette to 2 ml. So that's my working rate. So when I've already
done. I do the first count for the nematode. The first count, I pipette 2 ml. Quantify under microscope. How many do I get per 2 ml? I note down. Yeah. The most important thing again here is that you have your book, lab book. I know I have the sample here, 10. Let me take that is 4. Yeah, this is FM, label FM35. So I know where the sample came from. So I've just noted it somewhere, but I give it identity, right? So I label, this is FM35. I come to my book, put there FM35, right? Then I do the first count under microscope, note down how many. I go to the second count, how many. Then I go to the third count, then get how many. Why do I do the maybe 3 counts? Just to minimize the error, right? If we have very many replications, we minimize the error because we are human beings, right? Then after I've done the third count, then I get the average per 2 ml. Remember I was using the 2 ml, right? So from there, then I extrapolate. I want to assume that per 2 ml, maybe my average here maybe comes to maybe 205. Then I just say that if 2 ml is equivalent to 205 nematodes, I still don't know which diversity of nematodes which are here. Then about 10 ml, because that's the rate I decided to use, the standard rate. So then that is just a cross multiply 205 by 5. Yeah, by 5. So let's assume this is 1,000. Sorry, 1025. This is 1025 nematodes. So I've already known that in 10 ml, I have approximate of 1,225, right? Then from there, I will not identify all these nematodes because time. Let's say I have very many samples, like 20 samples or 50 samples or 100 samples. So sitting down to identify 1,000 nematodes is so time consuming. So what I do, I have another percentage, the standard percentage which has been used by scientists, 10 ml. I identify only 10 ml from this 205. So 10 ml, 10% of 1025, that is just 102. So I will identify 102 nematodes. So from there, this guides me. Now I come to the microscope identification. After that, I come here with my sample, FM, yes, this one. I come under microscope. I've already known how many numbers I'm going to identify. Yes, I come here and I've already known which number I should identify. Okay, okay. So I have my glass slide, I have the cover slip. Cover slip is here. Here, what is going to guide me is the number, right? So just focus on I want to pipette as many as possible so that I don't take much time pipetting so that I reach the number, the 202. So I've already pipette the bottom one, which I assume has got very many nematodes. I put it on the slide. And then I put a cover slip. So I've already put my cover slip. I come under microscope and put my sample there. So here is the most important. It's a very enjoyable part because you're going to interact with the real nematodes. Yeah. Why do we identify? Yeah. Okay. We identify for precise management maybe practices. Like you cannot just apply chemicals and you don't know what you're managing, right? So this is the most critical part of it, that from maybe a potato sample, which nematodes do you get? Like here, let us say we have already put the slide there. We want to see the nematodes. You just check. Oh, sorry. I would like you to see here as well. Okay, let me find them. I don't know. I think I have it. Yeah, yeah, yeah. I wanted to show you when potato cyst nematodes, when they hatch a juvenile, a J2 juvenile called Globodera rostokensis, the one which has been detected in Kenya. So I wanted to show you. Let me check this. It has disappeared. Yeah, there are some here. And I would also like you to come and check if indeed these are nematodes displayed on the screen. Yeah. I would see. Oh yeah, they're here. Yeah, I think this is the right one. Have you seen them moving? Are you seeing the ones who are still alive? They're moving. So there are some of the characteristics of some nematodes which are they move. There are some nematodes which move so fast. Okay, it's not. You see how that one is moving so fast? Let me bring some. Yeah. So this one, also there are some of the features. How would you identify a nematode? Like they have some features, very distinct features that enable us to either tell these are free-living nematode or this is a plant parasitic nematode. Yeah. So like this one, the one which is moving there, is a free-living nematode. So some of these free-living nematodes are so important. Like there are some which are viral vectors, like the I don't know, let me not mention the name. If somebody is not so much conversant with some kind of nematodes, we have the dory limes, the one which is now moving is a dory lime. So that one is a viral vector, though it's a free living. So it's important to know these viral vectors as well, so that when you're going for management, again, you can see how you can do the management. And some free living nematodes are so important as well, like bacterial feeders. Let me just find if I can just show you a bacterial feeder. Need to show you Lopodera. Anyway. Yeah, I would like to maybe one of you to come and see two. So like these are bacterial feeder, though it's not that clear. So some of these free living nematodes, like bacterial feeders, they normally indicate to us that the soil is very fertile, right? Like they are soil biodegraders. They biodegrade the humus. So nematodes can be used as a soil bioindicators. Yeah, like bacterial feeders. If I sample your farm and get many bacterial feeders, I know that soil was at a state of fertility. It's very fertile, right? But there are some nematodes, if I find them, they will just show me that their soil is infertile, maybe polluted, and very unstable. Yeah. So that's why it's very, very important to know which kind of nematodes are there. And when you are now applying your management, do you apply to which? Do you know the nematode you are managing? Yes. So maybe just to appreciate, you can check. If you don't see, you can fine tune. Yeah. You can again, yeah, maybe just move the mic. Yes.
Yeah, thanks, Calvin. The point here is not to get you to identify nematodes, just to show that there are different types, basically. We don't expect you to differentiate. And if you're doing nematode counts, you'd either send them to us or to crop nuts or something, basically. But it's just to show you that there's nematodes there and there are different types. But back to quantifying nematodes is if you've got a problem with your crop, you do an extraction, you see if there's nematodes there, parasitic nematodes, and then if you have a lot of parasitic nematodes, you can then pretty much debate, but assess and establish whether all those nematodes are part of the problem that you're seeing. But this would be done by us or by crop nuts or something like that, basically. But it's just to go through the process so that you're aware and that you can see what we do actually.
Yes, question.
Okay. Okay. Now, yeah,
question,
welcome
yes.
questions, right?
Now I've seen the nematodes swimming really well in water. Now in the soil medium, how do they move about?
How
they
do
move with the same speed as in water, or what happens?
How do they move? Like, let me say we have a nematode which is called potato cyst nematode. Yeah. When you plant your potato produces an exudate, like a liquid substance, which just make it know that there is a host here. So the potato cyst nematode now hatch. After they have hatched, the juveniles, the ones which are moving, they move towards the host and get into the pierce the root and get inside. They develop, I think you've been told, from the white, yellow, then finally it matures, and it comes out at the adult stage. So like potato cyst nematode is very specific to that kind of crop. But others, like bacterial feeders, they're just there, doing the soil, feeding on the bacteria, or some are enabling soil biodegradation. They biodegrade the substances in the soil. Yeah. Some like, yeah. So it depends on crop. Some nematodes affect some other crops, like root knot nematode. Yeah, there are several root knot nematodes which affect almost all crops. So they move, they move to find the host, towards the root. Yes. Yeah. Oh, how do they move? Yeah. They're yeah, and like serpentine, like, and again, when they move when there is moisture. Yeah. Okay, next question. Next question, yes, if there is any. Okay. I think if there's not any other question, then I would like you to go to the next table. Yeah, yeah. Mm.
Hi again, I'm Charlene, and I'm very glad to take you through culturing. When you are doing culturing for nematodes, I'm going to demonstrate how we do culturing for banana nematodes. Yeah, so that you can use them for inoculation, for setting up trials, experiments, and also to just have your own culture in case of anything. First, when you are culturing for banana nematodes, we usually use carrots or cocoyam. When working, you have to work under a fume chamber to maintain a sterile condition so that you can have sterile nematodes. So you need several equipment, the main thing being carrots here. You have your carrot. When buying, you have to be careful. Take a nice carrot, not too small, not too big, just nice one. You have your nematodes. Let's assume these are the nematodes. You set everything the way you see here, a pillar, a forceps. Inside the hood, you will have to have fire to sterilize some, yeah. Oh my God. So the first step is to make sure that you wash your carrots with running tap water to ensure that you remove excess dirt outside the carrot. Then you dry them with a paper towel. Take it to the hood. The first step, no, it's fine. I'll speak from. Let me speak from here. You can hear me? So, you are inside the hood. Now you want to work. Culture. Take your carrot, use this for support. That's enough.
Don't stab yourself.
Spray it with absolute ethanol, then flame it over the fire, just a little, not so much because you might end up thank you. Not so much because you might end up burning yourself, burning the carrot or even yourself. And also make sure that you don't put excess ethanol. Remember, it's absolute. After that, you do the first peeling. You can use a sterile paper like this, plain paper. Make sure you also sterilize this with flame and absolute. The rest of the things you autoclave. So you do your first peeling. The first peeling is to remove the outer parts. I can just demonstrate. Okay, thank you. So then put ethanol again, absolute, flame it, peel, continue peeling to remove such things. Make sure your carrot is now clean. Then you flame it again for the third time to shape it, make sure it's round. After that, and after every peeling, make sure you also sterilize this to avoid any contamination. After that, you can use either a blade or a knife, a small knife. I prefer using a blade. Sterilize it, cut into pieces, small pieces. Yeah, like that. Here we have our small round carrot disc. You see this outer part? This outer part is the one that the nematodes feed on. This one is just an excess. This is the main food. So even when you are peeling, make sure you don't peel deep into it so that the nematode can have the food, the necessary food. And you have your nematodes. They have settled here. Reduce water up to where you can normally see some white things. They will settle at the bottom, so remove the excess water. Then put antibiotic to sterilize the nematodes. We usually use streptomycin. After that, wait for an hour. Remove the excess water when the nematodes have already settled. Then after that, you do rinsing twice. You put water, wait for 30 minutes to 1 hour, remove, then again twice. After that, you've already quantified your nematodes. You know how many drops, how many nematodes are in a drop. So we want to inoculate. This is where you put your carrot, the round disc. Don't use your hand, please. You can use a small forceps, but in this case I don't have. So put it there. We usually inoculate 50 to 100 nematodes in a disc. So you've quantified, you know how many drops you put. Let's say 3. 3 are enough, like that. Yeah. There. You cover. Then remember to label what species of nematode you've cultured. For example, you write RS and the date. Those are the main important things. You will end up having something like this. Then you cover with a parafilm to avoid contamination and things like mites entering. Just, yeah.
Are the nematodes coming out of this?
Yeah.
Right, perhaps put that under the microscope and show people.
Yeah, here is an example of a radiolus culture. We can check under the microscope.
Let's go back. Give it to me.
So after that, you wait for a minimum period of 6 months, 6 weeks, then start monitoring your cultures to ensure they are clean, no contamination. Those that are contaminated, you remove them, and you can have your cultures, they have multiplied already. Then you can take them to the field, to the greenhouse, to inoculate for an experiment. How do you harvest from this? I'm going to show you. You have your water, clean water. First, you confirm if there are nematodes and if they're alive and they're enough. Then you put water on the cover, on the disc. You harvest using a pipette. When you are pipetting out, make sure you pipette in and out in the corners because they might hide there or, yeah. Then pipette it into a container like this. Go and quantify your nematode under the microscope. Then we go to inoculation part. Let's say this is a banana plant, tissue culture, and we want to inoculate. You make holes towards the root, just small holes. And then you've already quantified, you know how many nematodes you want to put in the plant. So just like that. Close the holes. And you monitor your experiments. So that's all. Any questions?
So again, we're not expecting that you will conduct this and you will do this. It's more of a demonstration of what we do as part of nematology, basically. But for example, if we're looking at, or Bayer, for example, might want to say, we really need to test our Velum against these two new products and a developing product. And so you may ask us, or you may ask JQuat, to actually assess the products on tomato, potato, banana, and to see how they work against the nematodes. So this is where inoculum is very useful. So you've got a ready supply of inoculum that you can inoculate plants, get them infected, and then you treat them with your different products, basically, to see whether you've got better efficacy for Velum against the others or others. And then you've got screening of different varieties. We may be looking at resistance of varieties. So each variety will be inoculated with the same number of nematodes, and to see if we've got resistance in this variety compared with this variety. And Charlene's showing you the culturing of Rhodophilus similis, which is the banana burrowing nematode. But it could be for potato cyst nematode, it could be for root knot nematode, it could be for others. Okay, and we have different techniques for culturing different species. But it just so happens that we do a lot of work on banana. So we've got cultures of the banana burrowing nematodes so that we can conduct our experiments on a regular basis without having to go to Kisi or Meru and harvest the field, and we've got mixtures of nematodes. So having pure cultures for inoculum is essential for us to do our work, basically. So we're not expecting you to return to Narok and start culturing nematodes, but it helps you to understand how we do our work. Yeah.
Yeah, question. Regarding your work here, have you done any work on potatoes, focusing on tolerance against nematodes or maybe resistance?
Yes, we have done. It's on this table. This part. Someone who does that work.
Yeah, we currently we are working on testing for resistance of different cultivars, different like right now we are using Shangi, Manitou, and some also varieties we received from SIP. So here we are testing for a specific resistant gene. It's called the H1 gene that is resistant to a pathotype in Kenya called Rostokiensis. Okay, so yeah, we do that too.
So all the varieties that we can get access to, we're inoculating them with nematodes to see whether we've got resistance or susceptibility.
That's all. Okay, thank you. If you have any questions, you can come back later. Like more questions.
Thanks, Charlene. Yeah, I think you guys are done now, actually.
Okay. Okay. They're finished. Okay. Okay. I would like now to maybe take you under maybe identification of the nematodes which have been extracted from that table. Okay. I hope. Okay. I'll just go with the team who are already here. Okay. So you have a sample which has been extracted there. Let me assume this is a sample which has been extracted there. It's now brought into the lab. This is now our lab. Sometimes these samples, when they extract them, they normally don't standardize them from the extraction. So they have different volumes. So when they come to the lab, you allow them to settle for, let me say, 30 to 40 minutes. The nematodes will settle down. So from there, you decide on which is the standard rate which you would like to use for your analysis. So here, you reduce the volume to, you can reduce to 10 ml. Yeah. So when you're reducing it to 10 ml, there are just some basic things which you should ensure that you do. You have your pipette, right? You reduce it, you ensure that your pipette is touching the walls of the Eppendorf tube. Pipette to the standard level, which is, I've just decided to use 10 ml. So I've already reduced to 10 ml. Now what I'm going to do the next is to go under microscope to know the quantity, how many are in that 10 ml, how many nematodes in that 10 ml. So I must always have my lab book. I have to label my sample, start with the ID. Let me say the label here is FM, maybe FM4. I have a column of, I have this column. I would like you to have that picture. I have the column there. I have ID, first count, second count, third count. Then from there, I will be doing extrapolation. I'll be just approximating how many are in that ml which I was using. So I've already done my labeling. I decide I'm going to use a pipette of 2 ml. So my first count, I pipette 2 ml, put under, there's a counting dish, a square counting dish, counting dish which can accommodate that 2 ml. You put the 2 ml there, count. The first count, how many do I get? Let me say I get 20, the first count. I count again, I get around, okay, yeah. So I've counted first. First, the basic thing here, I have to ensure that I shake my sample very well, uniformly. Then from there, I pipette 2 ml. So that whatever I'm pipetting will be a representative of kind of the nematodes there. So my first count there is 20 ml, 20 nematodes. The second count, again, I come and homogenize, just to ensure that
The nematodes are uniformly distributed in the falcon tube. After that, I pipette 2 ml, count there under microscope. Maybe I get 25. Again, I come to the last count, which is the third one. Homogenize, make it uniform, ensure the nematodes are uniformly distributed, pipette. Go and count again, I get around, let me say, 29. Then from there, I get the average part, 2 ml. I just do the average for 2 ml, then I say that I had a standard rate, which is 10 ml. Now, 2 ml has got, let me say the 2 ml has got 25. 2 ml is equivalent to 25 nematodes. About 10 ml. Now, here just cross multiply, that is 10 by 25 over 2. 25 by 5, that is 125. So in 10 ml, I'm able to get, I'm sure the 125 nematodes, at least, yeah, that's the approximation. So after I already have that count, sometimes you'll not be able to identify all of these nematodes, so you work with a certain percentage. So I say that I would like to identify only 10% of 125. So 10% of this is just 12. I'll identify 12 nematodes there. Yeah. So that is what now I come with to the microscope and identify. So I come with my sample. Yeah, that is done. Sorry. So I come under the microscope here, and now I have my sample. I have the slide here, glass slide. I have the cover slip. This is a cover slip which you use in the lab. Yeah. So I pipette. Now. So from here I'll just be focusing on, at least I would like to reach that number very quick. That 12, very faster. So I pipette the bottom one, as many as possible. Pipette. Put. After that, you put on the glass slide. Yeah. After that, then I just cover it so that the liquid doesn't touch the lens or the microscope. Then from there, okay, I put my slide there for identification. So now I would like you to appreciate these nematodes which are already under the slide, under the cover slip. And we first use this one. Yeah, these nematodes move. Let me put this. So why do we identify? Yes. Okay, here we have got the free living, we have got the plant parasitic nematodes. Huh? So why do we categorize some as free living, some as plant parasitic? If I start with the free living nematodes, you find that some of these free living nematodes, like bacterial feeders, they are nematodes which feed on bacteria. And you find that if you find them so many in your site or your farm, that will be very, very advantage to you, because these are soil biodegraders. Huh? Yeah, they just biodegrade so that the nutrients is available for your plants. But there are some free living nematodes which are also viral vectors.
like, you know, there is some viral vectors, viral diseases which affects potato mold virus. Are you aware of potato mold virus? Yeah. So some of these, some of the nematodes enhance the spread of those kind of virus to the crop. So it's also important to know which kind of those nematodes in your sample. Then again, there are some nematodes which may indicate that your soil is unstable or its soil is very polluted. So that's why it's very important to come here for identification. Then when you come to plant parasitic nematodes, that will now enable you to apply proper management. Let me say I sample from potato. I find very many juveniles of maybe Globodera, yeah. So it will enable me to apply if at all I'm going for this kind of chemical. I know that I'm going to suppress the number of Globodera, the cyst nematodes in the soil. So it's very important to identify. So I would just like you to now see the different kinds of nematodes here. No, I'm just, okay, I'm just showing you the nematodes which are in that sample. I don't know if there's one here. It appeared from the screen. It's there. Eh? So these are some of the vectors of the viruses. Oh, you can see it very. Oh, you can come and see under microscope here. Maybe just see. And this is just some nematodes. There's some of the free living organisms here, which are not so much important to the crops. They don't interfere with the crops. You able to see? Yeah, how would you tell some are viral vectors and which ones are not? So there are some, when you learn about the nematodes, about the classification, you'll be able to know some of the dory limes, some of the paratrichodoras, the trichodoras. Those are viral vectors. So this needs you to have that knowledge to know, is this a viral vector or not? But for plant parasitic, they have the stylets there, and they pierce and get into the roots. Yeah, the effect. Oh, this viral vector, yes. Mm. So there are some nematodes which are fungal feeders, some are bacterial feeders. The free living, they mostly fall under free livings. Yeah, when you see, you believe, huh? Yeah. No, there's so many, yeah. There's so many. They have just.
So you will count this one now, and then you move the slide or.
Okay, I will move my slide in a systematic way. Yeah. So I would like you to have that picture. So this is my counting. It is always a square. It has got square, small boxes. It's maybe 7 by 7. So I start from, when I choose where to start with. I can start from this end, go up to here, again come from this to the end in a very systematic way, so that I ensure that I don't miss any square. And when I reach the last box, If I don't reach the number which maybe I was identify 100, if I don't reach that number again, pipette, then I check again. Yeah, so you have to, yeah, that's why we use maybe that 10%. Huh? Yeah, so that you can do very many samples as possible. Yeah. Yes. Another question? Yeah. Okay, maybe if you don't have any question, you may just move to the next table. Most welcome. Yeah.
The first step you do, now that you have your carrot, you wash it through running tap water to remove dirt. Then you can dry it with a paper towel. What? Yeah. So, yeah, it's dry and clean. Take it to the hood where you've set your things, as I've explained. So,
And it has to be the lower tip of the carrot.
no, the whole carrot.
Okay.
Yeah, we'll get there. So you are in the hood. Remember, I told you everything is in sterile conditions to avoid any contamination. This is a forceps. Put it like this to support you and to avoid putting the carrot down. So then the next thing you do is you have your flame. You put ethanol absolute. Just pass it a little round. Don't too much, not over flame it, because it might burn the carrots. Then you peel, you do the first peel. The first peel is to remove the outer layer, this that you can see. Don't go deep, just the first. Then the second, sterilize this again with ethanol. Put it back here, this a rack that I use so that I don't put it on the surface. So you have your carrot, sterilize again with ethanol absolute. Flame it, just a little. And remember, when flaming, don't do like this. The ethanol might spread on yourself and you burn yourself. Peel again the second time to remove any dirt that you may, any layer, the first layer that you may have left. To shape to remove deep. You see, for example, here, there are these spots. You remove them. Then sterilize again, put it there, sterilize this for the third time. Now, when peeling for the last time, you are peeling to shape it, to make it round. After that, you can either use a blade or a knife. Me, I prefer using this. So you cut it into small pieces, round pieces. Not so big, not so small, just the right size. Put it in a petri dish, like this one. For big carrots, you can use a big petri dish. Please don't use your hand. Remember to have excess of this. You use it, put it on the petri dish, cover it, and wait for the nematodes. Now we are back to the nematodes. What you do with them. First, you have your nematodes in a container. This is an Eppendorf that I use. They are here. You wait for them to settle at the bottom. Then, remove the excess water on top. After that, you add antibiotic. Normally we use streptomycin. There you wait for them to settle for 1 hour. Remove the excess antibiotic. Then add water to rinse it. You rinse it twice. So you wait for a period of 30 minutes to 1 hour for the water part. After that, you have already quantified your nematodes. You know how many nematodes are in a drop. For example, yeah. Now we have the nematodes ready, you've quantified. In a carrot disc, we usually put 50 to 100 nematodes. So, you know how many nematodes are in your drop. Let's say for them to be enough, we have to put 2 drops. You see this outer part? That's where you place your drops. That's the part that the nematodes feed on. This inner part is not so much important. This is the main food, the outer part. So even when you are peeling, you have to be careful not to over peel it. Like you don't have to go deep into it. You wait for 6 weeks, then you can start monitoring your cultures that you. Wait, I forgot something. After you've inoculated, this is a very important part. Make sure you parafilm it. You seal it with a parafilm around to avoid contamination and things like mites entering into the disc. Also, label which species of the nematode you've cultured. For example, if it's RS, you write Rhodophylla similis. Then if it's any, and the date that you've cultured, you write it there. Then put it in a dark place. For Rhodophyllus, we put at a temperature of 28 degrees. For Pratylencus, we put at 25 degrees in an incubator, dark. Then, after a period of time, you have your cultures, they have multiplied, and you want to use them for an experiment, for a trial. You go and harvest them. How do you harvest them? You have your disc here. Just put clean water using a pipette. Put clean water on the disc. First, confirm if the nematodes are there and they're alive. Checking under the microscope, yeah. There is an example of a disc. I came with it. It's there. Maybe you can see it when you are done. Yeah, yeah, they are alive.
So I'm still stuck at the point where you're using antibiotic. What was it?
Sterilization.
Antibiotic was for sterilization?
Yeah.
And then ethanol was for what?
Oh, antibiotics specifically for the nematodes.
Okay.
Yeah, because we can't flame them, we'll burn them and they'll die. Yeah, the equipment and the carrot is where you use ethanol and flaming. Yeah. I'm not so sure about that point, but that's what is recommended. Yeah.
The antibacterial is only going to act on bacteria. What about the fungi and the lights?
For the mites, you cover with seal.
Fungus.
I think you just need to have sterile condition. Yeah, that's.
See, this one can be infected with fungus.
Yeah, sure, it happens. So, you have a question? Okay, so you have your harvested nematodes. Also, another point to note is when you are pipetting this, you make sure you pipette in and out in the corners. You know, when you remove some nematodes might get stuck around the corners and you might miss them. So after that, you have your inoculum ready. You've quantified under the microscope and you want to go and inoculate in a experiment.
So the reason why you put water there is so that you can be able to harvest the nematodes. So
Yeah.
you put water and then you harvest them, you put it into an inoculum.
Yeah, sure. Clean water. Then here is your banana. This is not a banana plant, just an example. Here is your banana plant. You want to inoculate. So when you're inoculating, you make holes. When making holes, you make sure you dig towards the roots. 2, 3, at least 2 to 4 holes, depending on how many samples you want to inoculate. So you have your pipette, measurements, just that, then cover, seal the holes, and you're done. Then you wait as you monitor your experiments.
So how long can it see the visual effects of attack?
Pardon?
If I put them today, after how long do I go and check the extent of attack?
For banana?
Banana or any other crop, now where you're integrating. Now I'm in the route, huh?
Yeah.
And I want to check whether my nematodes attacked
The roots, yeah.
The roots of the host.
Mhm.
How often do I look at it?
You are not, from what we do, we inoculate, then wait for a period of time, for example, 3 months. First, you have noted the number of nematodes you inoculated. Then, at the end of the experiments, maybe 3 months, depending on how long the crop takes to grow, you do termination. When you do termination, you go back to extraction. I believe you've been told about extraction. Yeah, then you can know how, if they have or if they haven't.
So it doesn't mean that if a crop takes 3 months, and I want to check my experiment after 1 month, I won't be able to do that. To see whether, no, by the 1 month, the crop has hair roots, the nematodes are inoculated in the first day. So meaning in the process of these hair roots and the roots for the plant growing, the nematodes are attacking. So I cannot be able to do it within a month.
It depends with your objective and the crop. Yeah, the best thing to do is just wait until the crop is fully grown up to the level you want. Then from there you can decide. But in between, it's advisable not to interrupt. Yeah.
And you recommend the soil to be moist and dry?
While inoculating? Dry. For example, for today, you don't water them. Then the next day, you can inoculate. And on that day, don't water them. You wait for, maybe you inoculate in the evening, very late, or the next day.
Thank you so much.
Okay.
Very much. We've come to the end of the practical session. I kindly request all of us to convene inside the tent. Kindly, those who are outside, karibuni ndani, to conclude the practical session and of course release our friends. How was it? Have you learned? You've learned a lot? Was it easy to understand? Are we somewhere in terms of getting sensitized on the nematode topic? Great. Okay. So, we are about to break for lunch, but it's important to note that after lunch, our friends will not be with us. So I thought we could all be in and possibly say a vote of thanks. Milka may also want to shake their hand and probably thank you, David, for being able to attend. We recognize the presence of our market development team. Good. So a lot from how to identify the symptoms in the field, to extraction, to counting. We can now say that when nematode is mentioned, we can visualize what it is, how the challenge can be, and the management. We are waiting still. Maybe I request our ISIPE IATA team not to pack now. Maybe we close this session and then they pack as we go for lunch, kindly. Ask them to come in. Thank you so much. Thank you so much, guys. Professor Dani, Dr. Solveig in absence, and Dr. Kanan, and the team, Miriam, and the strong technical team. Words are not enough to say thank you. We appreciate the expertise, the research angle, the knowledge that you've transferred to us today. And what we can promise you is that we are going to be the ambassadors of that topic at the farmer level. And not only ambassadors, but we will make sure that there is positive impact coming from today's engagement. Thank you so much. My colleagues have learned a lot. I'm sure there are a lot of questions to ask. This topic is the more you learn, the more you want to learn. We will discuss aside and see how we continue this engagement, how we make sure that some of the questions which are likely to arise as they interact with farmers are able to be answered correctly and with your support. Asanteni sana. We are going to have a 30 minutes lunch break, please. Our friends, IITA, ISIPE, we appreciate your presence and feel free to, have lunch with us, and thereafter you can leave at your own pleasure. The afternoon session will be very short. Actually, our planning on the next actions, especially next week going forward, when we are meeting as many growers as possible, preparing for this season. Thank you, guys. Enjoy your lunch. What's the time now? Yeah, can we be back at 2:35, please? Can we? Yeah, I've confirmed that lunch is ready, so straight ahead to the canteen and enjoy your lunch.